Hi everyone, it's Miss Lopez. These are the Chapter 4 notes, Cells, the Working Units of Life, continued. We've already talked about compartmentalization for the biochemical reactions, as well as the difference between prokaryotic cells and eukaryotic cells. We've talked about the endosymbiotic theory, and we've also talked about how the size and shape of a cell is going to affect how the cell functions. What we're going to do in these last set of notes is look at the subcellular compartments. In other words, we're going to be looking at the organelles and components of the cytoskeleton of the cells. As a reminder, the two types of cells we are looking at are prokaryotes and eukaryotes. And the key difference between them is that prokaryotes do not have membrane enclosed compartments. And that's the key thing, is that they're membrane enclosed. So that doesn't mean that prokaryotes can't do biochemical reactions or compartmentalization. They just don't have specified membrane-bound organelles in which these are going to happen. Eukaryotes do have those membrane-bound organelles, and one of the most easily distinguishing features between the two is the presence of the nucleus. Here's a representation of a typical animal cell. You'll notice many of the organelles that you studied in your Quizlet set are labeled on this diagram. Here is a typical diagram of a plant cell. And many of the organelles are labeled for you. Some of the key things to look for in trying to distinguish between an animal cell and a plant cell include the presence of a cell wall, the presence of a large central vacuole, the presence of plasmodesmata, and the presence of a chloroplast. You can use these key features to distinguish between a plant cell and an animal cell. We're not going to take too much time going over the specifics of what each organelle does, except for a few examples that might be new in AP biology. However, it is very important that you know how these organelles work together to accomplish a particular function for the different types of cells. Now, in this example, we're going to look at how cells can work together to secrete a substance from the cell. In starting this process, we begin at the nucleus, where the DNA of the cell is contained. The DNA is going to be expressed in the form of proteins. And so the first thing that's going to happen in this cell is that this DNA is going to get converted into proteins. So the DNA will be transformed to mRNA, and we'll talk about that process in January. And then the mRNA is translated or turned into proteins in the ER, specifically the rough ER. Then the ER takes those substances, those proteins, and packages them into small vesicles, where they will then travel to the next organelle, which is the Golgi. At the Golgi, those vesicles, those proteins are modified and then packaged into separate vesicles that then travel to the plasma membrane, where they fuse with the plasma membrane, and then the substances are released outside of the cell. So let's write that process down. For cells that are going to secrete specifically a protein, now they can secrete other substances, but for a cell that's going to secrete a protein, we're going to start at the nucleus, where the DNA or genetic material of the cell is kept. The DNA will be converted into proteins, two processes called transcription and translation, which we'll go into more detail about in January. For now, just know that the DNA is converted into proteins at the ribosomes, specifically the ribosomes on the rough ER. From there, the proteins, which were encoded from the genetic information, get sent to the Golgi. At the Golgi body, The proteins are then going to be modified. In other words, they're going to undergo those final levels of protein folding, the quaternary levels of protein folding, and then they're going to be packaged up into vesicles. Those vesicles then travel to the plasma membrane where they fuse with the membrane so that the particles can be released. It's important to take note that with the vesicles, you must include the word fusing with the plasma membrane. This was an example on an FRQ where students did not earn credit if they just said that the vesicles travel to the plasma membrane. You've got to indicate what happens to the vesicle once it gets to the plasma membrane, and in this case, the word was fusing. So the vesicle fuses with the plasma membrane, and then the substances are released from the cell. Now, this would be very similar in a process where it is secreting a lipid of some kind. The difference between that is that in the case of a lipid that's being secreted, instead of coming from the rough ER, the ribosomes on the rough ER, we would have the substances being made in the smooth ER. So I'm going to add that underneath. Now let's take a look at another specific example of how structure affects functions. We're going to look at erythrocytes, also known as red blood cells. Sometimes I abbreviate red blood cells as RBCs. So red blood cells have one sole function and purpose in their limited 7-day lifespan, and that is to transport oxygen. Because they only have one function in transporting oxygen, erythrocyte structure is very different from that of other cells. They lack all organelles, nucleus, endoplasmic reticulums, mitochondria. They have nothing in them except the protein hemoglobin for transporting oxygen. They still have a plasma membrane. The plasma membrane is not an organelle. And as a result of lacking all organelles, erythrocytes have a very peculiar shape. This indentation you see right here is formed because the normally full cell has been emptied of a lot of its volume in the organelles, and so it kind of creates what we call a concave shape. Now, since this cell is lacking all organelles, if you did analysis on it, you would not see those membrane-bound organelles. And you would do the analysis by a staining technique. One last example of how structure affects function is cells whose function it is are involved with movement. Now, if a cell is involved in movement, there's several things that it's going to need in order to be able to move. This could be something like flagella, cilium, pseudopods. In any case, regardless of how the cell moves, there's going to be a lot of energy involved in this process because movement is a very energy expensive process. So one of the things you're going to notice in these cells is a lot of mitochondria. You'll also have many of the other typical types of organelles involved, but mostly you're going to notice a lot more mitochondria, and you're going to notice some type of organelle that's involved in movement. Those are just a few examples. What other examples of cells could we talk about, or could College Board ask you about, in terms of how structure affects function? Here are some to think about, but please remember this is not the exhaustive list. The College Board may have something that I didn't think of. They could be cells that specialize in secreting ions. They could be cells that specialize in structural support. There could be cells that specialize in detoxifying or waste removal. They could be cells that specialize in absorbing nutrients. They also could be cells that specialize in passing chemical signals. So keep the structure of the cell in mind when you're trying to figure out its function. You want to look at what are the subcellular components. but also take into account that the size and the shape of the cell can also aid in its function. Now we're going to focus on the structure of the cytoskeleton. The cytoskeleton supports and maintains the cell shape. It holds the organelles in positions, and it also moves organelles such as vesicles. It is involved in cytoplasmic streaming, which is moving the cell itself, and the cytoskeleton interacts with the extracellular structures to anchor the cell in place. The three components of the cytoskeleton include microfilaments, intermediate filaments, and microtubules. Let's start with microfilaments. Microfilaments help a cell or parts of a cell to move. They determine the cell shape, and they're made from actin, monomers that attach to the plus end and detach at the minus end of a filament. So here is the actin filament, and you can see that it's got a negative end and a positive end to it. These little spheres represents different proteins, and down here you can see a colored fluorescent micrograph or picture of the actin filaments. Dynamic instability is the idea or the process that the filaments can shorten, become more detached, or lengthen more assembly. This allows for quick assembly or breakdown of the cytoskeleton. In muscle cells, actin filaments are associated with the motor protein myosin. Their interactions result in muscle contraction. Motor proteins, molecular motor, are any proteins that cause the movement within the cell. And so one of the important ways to distinguish between the three different types of cytoskeletal elements include whether or not that cytoskeletal element displays that dynamic instability. A simpler way to think about this is dynamic instability is talking about whether or not the cytoskeleton element is able to grow and shrink, or does it remain in the same position and the same size the entire length of the cell's life. Intermediate filaments are at least 50 different kinds in 6 molecular classes. They are tough, rope-like protein assemblages. They are more permanent than other filaments, and they do not show dynamic instability. This makes them very good for anchoring cell structures in place, and they resist tension, maintaining the rigidity of the cell so that the cell doesn't collapse on itself. Here you can see the colored micrograph of the intermediate filaments. They are stained green, and they do not display that dynamic instability, so they maintain their shape and they maintain their size and act as good anchors in the cell. The last cytoskeletal element that we're going to talk about are microtubules. These are the thickest cytoskeleton elements. They form a rigid internal skeleton for some cells or specific region, and they act as a framework for motor proteins to move structures in the cell. In this picture, the microtubules are stained in blue, and you can see that they have a negative and positive end, meaning that they do show dynamic instability. So these can grow and shrink depending on the needs of the cell. Microtubules are made from dimers of the protein tubulin. Chains of dimers surround a hollow core. They have that negative and positive end that show dynamic instability, and polymerization results in a rigid structure. Depolymerization leads to its collapse. Microtubules form an internal skeleton for movable cellular appendages, such as cilia and flagella. You're probably familiar with microtubules in the process of cell division. When the chromosomes are lined up in the middle of the plate, it is the microtubules that play tug of war with the chromosomes to make sure they line up in the middle of the plate during metaphase. We will talk more about the cell division process as well as the microtubules and their role in the cell division process in January. Now let's talk about the external parts of the cell, starting with the cell wall. In plants, the cell wall is a semi-rigid structure outside of the membrane. The fibrous component is the polysaccharide cellulose, which is made of beta-glucoses. The gel-like matrix contains cross-linked polysaccharides and proteins. So here's a micrograph or a picture of the cell wall. So you can see the membrane right along this darkened edge here. And then you can see the cell wall is a separate component. Now keep in mind that this is also going to be found in bacteria. A cell wall is going to be found in bacteria, but instead of cellulose, it will be made of peptidoglycan. You also find cell walls in the fungi group, but instead of cellulose, they are made of chitin. The plant cell wall has three major roles: provide support for the cell and limit its volume by remaining rigid, act as a barrier against infection, and contributes to form during growth and development. Adjacent plant cells are connected by cell membrane-lined channels called plasmodesmata. These channels allow for movement of water, ions, small molecules, hormones, and some RNA and proteins. Outside of the cell is known as the extracellular matrix. Many animal cells are surrounded by this extracellular matrix and can include the fibrous component made of collagen. Cell junctions are specialized structures that protrude from adjacent cells and glue them together. They include tight junctions, desmosomes, and gap junctions. Tight junctions prevent substances from moving through the spaces between cells. Desmosomes hold cells together but allow materials to move in the matrix. Gap junctions are channels that run between the membrane pores in adjacent cells, allowing substances to pass between cells. Gap junctions are very similar to the plasmodesmata we saw in plants. The difference is gap junctions are in animals, plasmodesmata are in plants. So you can see here the tight junctions holding the cells together. They also prevent the passage of substances between the cells. So tight junctions hold the cells together tight. Desmosomes, as shown here, provide links in between the cells for the movement of substances, but also aid in giving the cell strength. Gap junctions are simply channels in between the cells where there can be an exchange of ions or substances. Thank you guys so much for watching. I hope this video was helpful. I'll see you guys next time.